Hello everyone, welcome to my channel. My name is Laha. I am an assistant professor of Department of Botany, Chandanagar College. Today I will be giving a seminar on marubin and alternative to allergy treatment as a part of online refresher course in biological sciences being organized by UGC-HRDC, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. So, I will begin by talking about allergy and allergens. Allergy is a IgE mediated type 1 hypersensitivity reaction, and this occurs when an immune system reacts to an allergen. Allergen are some glycoprotein or proteinaceous compounds that can elicit an allergic immune response. There are various symptoms of allergy. It can have local symptoms like asthma or atopic dermatitis, or some systemic allergic reaction like anaphylaxis. Allergy can be caused due to various reasons. One can have allergy from house dust mite allergen, or from food allergen, or from pet allergens. Even some of the climate change and everyday exposure to smoking and pollutions and viral infection can aggravate the symptoms of allergy. Globally, 10 to 13% of the world population are suffering from one or the other type of allergy. By 2050, the World Health Organization predicts that 50% of the people worldwide will be suffering from asthma. And according to a hospitalization-based survey in India, nearly 15% of the people have developed atopic asthma, and 20 to 30% of the population are suffering from allergic rhinitis. So, how does the allergy takes place? During the fast exposure of the allergen, the Th2 cell stimulates the B cell to form a IgE secreting plasma cell. These secreted IgE molecules goes on to bind with the Fc TAR1 receptor present on the surface of the mast cells and the basophils. Then this second allergen exposure takes place, which leads to the cross-linking of the IgE molecules, which leads to the phosphorylation and activation of the tyrosine kinase lin, which then activates the protein kinase C. It then activates the downstream phospholipase D by phosphorylation, which causes the degranulation of the mast cell. The PKC also activates the NF kappa beta, which leads to the production and release of cytokines. The lin also activates MAPK, and which goes on to activate phospholipase A by phosphorylation and leads to much more production of leukotriene and prostaglandin. Thus, due to the cross-linking of the IgE receptor molecules, the release of various inflammatory mediators or like vasoactive amines like cytokines, leukotrienes, and prostaglandins takes place. It results in a clinical manifestation of various allergic symptoms like rhinitis, urticaria, etc. In order to diagnose the cause of allergy, the medical history has been asked by the physicians, and several in vivo and in vitro tests ensuing oral food challenge test, the elimination diet test has been performed. In the in vivo test, the skin prick test or the skin prick by prick test has been performed. In the in vitro test, the total serum Ig has been estimated. The basophil activation test has been performed. However, from this diagnosis, various sorts of medicines have been prescribed by the physicians. It includes inhaled corticosteroids, leukotriene receptor antagonists, and H1 antihistamines. All of the medicines which are used to treat allergic asthma and allergic rhinitis have several side effects. Like H1 antihistamine can cause cardiac events, leukotriene receptor antagonists can have neuropsychiatric effect, inhaled corticosteroid can lead to diabetes or other respiratory infection. Even the inhaled corticosteroids can cause neuropsychiatric problem. So in order to get rid of the side effects of those particular medicines, nowadays scientists are looking into various natural products to combat the allergy, and one such particular chemical compound is flavonoids, which is available in many plants and has been reported to have anti-allergy properties. Our interest is in marubin, which is a diterpenoid lactone which can be obtained from plants of Lamiaceae like Marubium vulgare. This particular chemical compound marubin has been reported to be antioxidant, anti-genotoxic, immunomodulating, vasorelaxant, and anti-diabetic effect. It also has a high stability, low turnover, and little catabolism, which are essential qualities to develop nutraceuticals with significant economic value. Nowadays, a system immunology approach is being utilized, which is an interdisciplinary field which uses the immune system communication molecules and tries to understand the immune system signaling pathways. But in order to curtail down the cost of the wet lab experiments, computational biological techniques have been utilized to study the interaction of FcR1 receptor and various other drugs. Like for example, the Discotope2 servers, Elipro servers have been utilized from the data of 25 mite allergen sequences to generate a 64 consensus conformational beta B cell epitopes. Even Elipro, FireDoc, PayFol3, PassDoc, NetCTL 1.2.2 can be used to identify the potential T cell and B cell epitopes in Epistem-Barr virus. So the objective of our study is in silico investigation to discover any interaction between the high affinity Ig receptor FcR1 and an anti-allergic substance, which is marubin. In order to perform the experiment, the following methodology was utilized, where the structure of the FcR1 receptor was downloaded from the protein data bank, and the structure of the anti-allergic molecule marubin was downloaded from PubChem. And then the marubin was subjected to in silico analysis of drug likeness parameter in the online server AdmitLab 2.0. After that particular marubin and FcR1 receptor were subjected to molecular docking with the help of the software Autodoc software version 4.2.6, and the molecular simulation was performed using this CHARM 27 force field and GROMACS 2021.2 utility. During this particular ADMET analysis, the data is visible here. Here you can see various parameters have been analyzed, and these on the right hand side, these are the data which have been obtained from the ADMET Lab 2.0 analysis, and it has been found out that marubin has qualified the GSK rule, Lipinski rule, and Golden Triangle rule of ADMET Lab 2.0. Even in order to qualify as a permeable in vivo drug, it has marubin has qualified the CACO2 and MDCK2 permeability test. It has been confirmed by the ADMET Lab to test that the marubin would not cause any sort of damage to the liver, as it has qualified the drug induced liver injury test too. And also marubin compiles with skin sensitization rule, the non-biodegradable rule, and the Seor-Campbell criterion. Thus, it confirms that marubin can be safely used for the future in vivo clinical trials. Then the marubin and FcR1 receptor were subject to molecular docking, and its base dock pose were subjected to molecular simulation. And here you can see the time evolution of the root mean square deviation, or the RMSD graph of the backbone conformation of the dock complex through 1 nanosecond. And this is the 3D diagram of the marubin interacting with FcR1 receptor, and this is the 2D depiction of that particular interaction. And from this thing we can see that there are two conventional hydrogen bond, one carbon hydrogen bond, and one pi alkyl bond has been formed, and the binding energy has been observed to be minus 7.41 kilocalorie per mole, and inhibition constant of 3.69 micromole. These conventional hydrogen bonds, which have been created between tyrosine 129 and valine 118 along with the human high affinity Ig receptor or FcR1 receptor, actually provides the stability to the complex. Even the molecular dynamic simulation, or the RMSD graph which I have shown you earlier, did not exhibit any sort of structural changes. And during this simulation and the docking, the bonds were found between the residues 118, 129 and 131. And it must be mentioned that this particular three residues, 118 residue in the D2C strand and 129 and 131 residue in the C prime E loop of the FcR1, are crucial for IgE binding. Thus, according to our investigation, it can be seen that marubin is getting engaged with the binding with the FcR1 receptor at that particular three residues. Therefore, it will successfully block the downstream allergy pathway by stopping IgE from activating the human high affinity IgE receptor, or the FcR1 receptor. Thus, from our study, we can conclude that human high affinity Ig receptor, or FcR1 receptor, acts as a key regulator in mast cell activation during allergic responses. However, our in silico docking study demonstrated the significant inhibition of Fc eta R1 receptor by marubin, thereby suppressing the interaction of Ig with Fc eta R1 receptor, and it thus highlights that marubin is a promising anti-allergic agent in the future. However, before implementing this marubin as a medicine, various other clinical trials must be performed to gather substantial evidence about its safety. Now coming to the references, these are some of the references which I have consulted while designing and performing this particular work. And I would like to acknowledge my PhD supervisor, Dr. Rajiv Bandyopadhyay, Professor and Head of Department of Botany, the University of Burdwan, the Honorable Vice Chancellor of University of Burdwan, and several other faculty members of the university for giving me the chance to perform the experiment. And also I would like to acknowledge our principal, Dr. Debashish Sarkar, and Director of Public Instruction, Professor Jasir Rai Choudhury, for giving me the chance to perform this experiment in collaboration with the University of Burdwan. And I would like to acknowledge all other people for guiding me through my journey of research work. Thank you all.